On this week's Arabidopsis Research Roundup, I'm joined by Giles Johnson from the University of Manchester, and we're going to be talking about a recent paper just published in Plant Physiology, the title of which is FUM2, a cytosolic fumarase is essential for acclimation to low temperature in Arabidopsis thaliana. So thank you very much, Giles, for joining us today and discussing this paper. So if you can give us a brief overview, that would be great. Thanks.
Okay, so this is part of a study which started as a BBSRC special initiative looking at ways of improving photosynthesis. So we were interested in the possible role of fumarate as a storage compound for photosynthesis, as a carbon store. And the idea originally was we were interested in how could you improve carbon storage. However, along the way, what we actually found was that fumarate seems to be preferentially accumulating at low temperature. So we looked at plants which weren't able to do this. So it's previously been shown that plants which lack a cytosolic form of the enzyme fumarase are unable to accumulate high concentrations of fumarate across the day-night cycle. And when you knock out the fumarase 2 gene, which is a cytosolic form of that protein, then what you find is you lose all accumulation of fumarate, but you also lose the ability to acclimate photosynthesis to low temperature.
Okay.
So we're looking at how the photosynthetic capacity of plants changes when you transfer them from high temperatures, at moderate temperatures, 20 degrees, to low temperatures, 4 degrees. And we found that in wild-type plants, the photosynthetic capacity increases over a period of about 5 days. In plants which lack FUM2, you completely lose that ability to increase photosynthetic capacity.
Okay. So can you outline a few of the experiments you did within the paper to find this out?
So the way we present the story in the paper, we started by using analysis with gas chromatography, mass spectrometry, GCMS. Mm-hmm. And what you find is that looking at the response of plants to low temperature, fumarate stands out as being one of the most important compounds using multivariate analysis. We found that using more directed, focused analysis, we found that the concentration of fumarate, we could confirm that the concentration of fumarate was increasing. We then did analysis using, we did a transcriptomic analysis to show that in the wild type, you don't actually get, when you transfer plants from high temperature to low temperature, you get changes in lots and lots of genes, but you don't particularly get changes in photosynthetic genes. Photosynthetic genes tend to be quite constant. Mm-hmm. But in the mutant, which is unable to respond to low temperature, genes associated with photosynthesis all seem to go down, or a lot of them seem to go down in terms of their expression level.
Okay.
Then we went on to measure, using physiological measurements of gas exchange, we were able to show that although those genes are going down, the protein concentration associated, the activity rather of photosynthesis stays very much the same. But in the wild type, where the gene expression is not changing, the activity of those enzymes is going up.
So maybe a more general question: why is it that do we know why fumarate or fumaric acid is a store in cold? Why is it involved in cold, and why not another component of the TCA cycle?
I think the simple answer is always no. That we're trying to find that out.
Right.
We obviously have lots of ideas about this, and we're trying to get funding at the moment to explore some of those ideas. We don't think it's simply that fumarate is acting as a store of carbon. Okay. So it's not, for example, you can look at other conditions which result in an increase in total leaf carbon. So, for example, you can do experiments where you increase the light intensity, you do more photosynthesis, mm-hmm. you fix more carbon, but you don't get an increase in the accumulation of fumarate. Okay. You can do the same with high CO2. We get the same effect, but high CO2 doesn't increase fumarate accumulation. So it's not simply a dumb store. It's not simply that some of the carbon is being diverted in that direction. It's specific to cold conditions.
Okay. So do you know why it's specific to cold, that particular molecule?
Well, that's as I say, that's something that we're hoping to continue working with in the near future. Okay. I think it can't be taken in isolation. You have to see it in the context of whole metabolism, and that's why the approaches we're using include looking at modelling of metabolism. So we're collaborating with people here in Manchester, mm-hmm. Using flux balance analysis, for example, trying to understand how metabolism is being perturbed by low temperature, and how the accumulation of fumarate modulates that.
Okay. And in conditions of no cold at all, so in normal growth, these FUM2 mutants, do they look completely normal? Do they grow normally?
Yeah, I mean, we're not able to pick up any kind of phenotype at 20 degrees. So you measure although the capacity for photosynthesis is slightly lower, the actual rate that's achieved under our growth conditions is the same as in wild type. So we've looked at how photosynthesis varies through the day. They're indistinguishable. They're indistinguishable in terms of their overall growth rate as well.
Okay, I see.
So it's only when they're transferred to 4 degrees that we start to see this phenotype.
Okay. And I suppose this is all within a larger goal of trying to find, and you highlight this in the discussion, so trying to find the cold sensor, let's say, within the plant. So do you have any indications, the direction where that cold sensor might be? Are you looking to find that yourselves, or are you in collaboration with people?
We would very much like to find out what the mechanism is of cold sensing. I mean, in terms of... So one thing to bear in mind is when you talk about cold acclimation, then we're talking about quite a lot of different things which are going on at the same time, and it's almost certainly the case that there's more than one sensor. Mm-hmm. So obviously an aspect of this will be cold hardening, so resistance to frost.
Yeah.
And in terms of that aspect, so genes which are known to be associated with cold hardening, so some core genes, for example, they're still showing a responsiveness to cold. So it's not that we've knocked out all ability to sense low temperature.
Yeah.
There clearly are other things going on.
Right.
Although in some cases the expression of those genes does seem to be perturbed in our mutants.
Okay.
We think this is more particularly linked to the sensing of changes in metabolism and of optimizing metabolism to suit the conditions that the plant's experiencing.
Okay. Cool.
Previous work, earlier work, there's some really nice work from I guess 10, 15 years ago, something like that. From Vaughan Harry, Mark Stitt and others, who they suggested that there was actually the response to low temperature was probably linked to, amongst other things, phosphate and the amount of free phosphate. So at low temperature, phosphate tends to get tied up in sugar phosphates, and the availability of free phosphate can become limiting to photosynthesis. Mm-hmm. We don't think that's a signal that's controlling acclimation in this case, though, because in our case, in the mutant, we've actually got higher concentrations of sugar phosphate, lower free phosphate, and then we're getting in the wild type. So we think it does, certainly the synthesis of fumarate does free up some phosphate. I think that's likely the case, but we don't think the phosphate itself is a signal for acclimation.
Okay.
Which is what's maybe been suggested before.
Okay, as with anything, when you're talking about a metabolic flux, you look at one thing and the other thing changes, and it's obviously an extraordinarily complex situation. So teasing out one particular component is obviously extremely difficult.
Which is why I think that the way forward is to apply modelling to this. So,
yeah.
I like to compare it to one of those sort of stress balls that you squeeze, one of those stress relievers you squeeze.
Yeah, okay.
When you squeeze it, then things come bulging out in other directions. So I think that's what's happening to metabolism when we perturb it in this way. So trying to work out, I think in order to work out what the important factors are, you need to try squeezing it in other places. Yeah. So that will be our major strategy, will be to use different mutants in different parts of, particularly different parts of the organic acid pathway, to see how they're perturbed, and then combine that with modelling to try and understand the whole dynamics of the system.
Okay, excellent. Well, it sounds like there's some exciting ideas there. Now, obviously the funding needs to come in for you to study that, as ever, of course. But
indeed, yes, of course.
Well, okay. So thanks very much, Giles, for joining us today. That's great.
Okay.